the today's session, we are going to talk about Alzheimer's disease, its pathology and treatment. Now, Alzheimer's disease is a progressive neurodegenerative disease. It is termed as to be the neurodegenerative disease because there is degeneration and death of neurons. And it is a disease of old age. It is a disease of old age because it normally affects adults over the age of 65 years. It is the most common cause of dementia, characterized by impairment of memory and cognition. Now, impairment of memory refers to inability to recall, especially the short-term recent memories. There is forgetfulness and getting lost usually. And impairment of cognition refers to inability to think, plan, pay attention, inability to judge, inability to take decisions. So it is a disease that is characterized by dementia, and dementia is the impairment of memory and cognition. Now, these are the two diagrams I have drawn over here. This is the brain of a healthy person, and this is the brain of a person suffering from Alzheimer's disease. Now, as it is clear from the diagram, there is atrophy or the shrinkage of brain because of the Alzheimer's disease, because of the neurodegeneration and death of the nerve cells. Now, because of the progressive neurodegeneration, certain microscopic changes are observed in the structure of brain. And it is the hippocampus and the cerebral cortex of the brain which is affected first in the patient of Alzheimer's disease. Now, as it is clear from the diagram, there are certain characteristic ridges of the brain. These are the characteristic ridges of the brain, and these ridges of the brain are termed as to be the gyri. And because of the shrinkage of the brain, these gyri they become narrow. So because of the shrinkage of the brain, these gyri they get narrower, and sulci are the grooves between the gyri. This is a gyri, this is the next gyri. The groove between the gyri is termed as to be the sulci. And because of the shrinkage of the brain, these sulci they get widened. There is widening of the grooves, there is widening of the sulci. This is because of the shrinkage of the brain. And apart from this, the ventricles which are found in the brain, they get severely enlarged. They get severely enlarged because of the shrinkage of the brain tissue, because of the neurodegeneration and the death of the nerve cells. Now, there are three hallmarks of Alzheimer's disease. First is the beta amyloid senile plaque. In the Alzheimer's disease, there is excessive generation of beta amyloid protein. These are the big proteins, and they're highly sticky. They aggregate together to form senile plaques, and these senile plaques they block the neuronal junctions. And because of the blockage of the neuronal junctions, there is disruption of the neurotransmission. And because of the disruption of the neurotransmission, there is neurodegeneration followed by the neuronal death. And apart from this, the second hallmark of Alzheimer's disease is the tau protein. In Alzheimer's disease, there is excessive phosphorylation of tau protein. Now, this tau protein is responsible for stabilizing the microtubules within the nerve cells. And phosphorylation of tau protein results in the detachment of tau proteins from the microtubules. And these phosphorylated tau proteins, once they have detached themselves from the microtubules, they aggregate together to form neurofibrillary tangles. So, second hallmark of Alzheimer's disease is the formation of neurofibrillary tangles within the nerve cells. And because of the formation of these tangles, there is destabilization of microtubules, and which disrupts the normal intracellular transport mechanism of a nerve cell. The third hallmark of Alzheimer's disease is the dysfunctional cholinergic neurons, and these dysfunctional cholinergic neurons they synthesize less amount of acetylcholine, and there is less release of acetylcholine in the synaptic cleft, and because of which there is reduced cholinergic transmission, and because of the again reduced cholinergic transmission there is neurodegeneration and death of the neurons. Now, one by one we are going to study all the three hallmarks of Alzheimer's disease, which are basically responsible for the progression of Alzheimer's disease from mild to severe. Now let's talk about the first hallmark of Alzheimer's disease, formation of beta amyloid protein. Now we'll talk about the amyloid precursor protein, and this is a nerve cell membrane. It is a part of nerve cell membrane that forms a synapse. As we all know, synapse is a junction between the two nerve cells where the neurotransmission takes place. So this amyloid precursor protein, in this diagram I have shown this with the green color. It is a part of the nerve cell membrane, and this amyloid precursor protein, it is required for the nerve growth and the repair. Now this amyloid precursor protein, it is broken down into fragments, and these fragments are removed off or disposed of from the body by two pathways. One is the non-amyloidogenic pathway, other is the amyloidogenic pathway. In the non-amyloidogenic pathway, the two enzymes, namely alpha secretase and gamma secretase, they break amyloid precursor protein into small soluble fragments, and these small soluble fragments are disposed of easily from the body. Whereas when we talk about the amyloidogenic pathway, the two enzymes, namely the beta secretase and gamma secretase, they break amyloid precursor protein again into three fragments. Now beta secretase, it breaks the amyloid precursor protein in such a way that a comparatively larger fragment is produced, and this larger fragment of amyloid precursor protein is termed as to be the beta amyloid protein. And this beta amyloid protein, it is made up of 36 to 43 amino acid. So comparatively this is larger, this fragment is larger in size, and this protein, it is also very sticky, and since it is sticky, it forms oligomers, it aggregates if it is not removed from the body. Now this beta amyloid protein, it is removed by the process of phagocytosis. Phagocytosis induced by the microglial cells. It is removed by the receptor mediated internalization by astrocytes, and it is also broken down by the protease enzyme. Protease enzyme, which is termed as to be the neprilysin. So these are the methods which are responsible for disposing of beta amyloid monomer protein from the body. Now what happens in the Alzheimer's disease is this, that there is excessive formation of beta amyloid proteins, and there is reduced disposal of beta amyloid proteins, and because of which there is accumulation of beta amyloid proteins in the neuronal junctions. And this beta amyloid protein, once it accumulates in the neuronal junctions, it aggregates and it forms beta amyloid senile plaques, which block the neuronal junctions, disrupting the process of neurotransmission, resulting in progressive neurodegeneration followed by the neuronal death, which is one of the main causes of Alzheimer's disease. Now, a second hallmark of Alzheimer's disease is the tau protein. Tau protein is a protein that is required for the stabilization of microtubules in the nerve cells. Now, as we all know, the function of microtubules is the intracellular transportation. These microtubules, they are required for the transportation of organelles, like for example mitochondria, then for example vesicles within a nerve cell. They are also required for the transportation of biochemical, they are required for the transportation of nutrients, waste products within a nerve cell membrane. So this I have shown here with a diagram. These are the microtubules, and this is the tau protein. This tau protein, it is responsible for the stabilization of microtubules. Now, as we have already discussed, that in Alzheimer's disease there is excessive formation of beta amyloid proteins. Now, these beta amyloid proteins, they activate intracellular kinase. Now, activation of kinase causes phosphorylation of this tau proteins. So, this tau protein, which here I have shown in the red color, this tau protein, it gets phosphorylated. And once it gets phosphorylated, it detaches itself from the microtubules. Now, when the tau protein detaches itself from the microtubules, these microtubules, they get destabilized, and further there is crumbling of the microtubules. So there is a disruption of the intracellular transport within a nerve cell. So, activation of intracellular kinase. Kinase phosphorylates the tau protein. Phosphorylated tau detaches itself from the microtubules and it aggregates within the nerve cells. Now, these aggregates of tau proteins, they are termed as to be the neurofibrillary tangles, and these neurofibrillary tangles, they are found within the nerve cell. Now, these because of the formation, because of the detachment of tau protein, the microtubules, they destabilize and they collapse. Here I have shown that because of the removal of the tau protein, these microtubules, they have become destabilized and they finally collapse. And because of which intracellular transport is disabled, and because of the because this intracellular transport is disabled, there is neurodegeneration followed by the neuronal death. So this is the second very important hallmark of Alzheimer's disease. And the third hallmark, as we have seen earlier, is the dysfunctional cholinergic neurons. Now this slide shows a very important diagram. In the diagram are shown two neurons. This is a synapse. This is the presynaptic neuron. This is the postsynaptic neuron. Now, this is a part of the nerve cell membrane that forms the synapse. And as we have discussed earlier, amyloid precursor proteins are found to be present in this nerve cell membrane. And amyloid precursor protein, they break to produce the beta amyloid proteins by the amyloidogenic pathway. And in Alzheimer's disease, there is excessive formation of beta amyloid proteins which are found to be sticky. And these beta amyloid proteins, they aggregate together to form the oligomers. Oligomers, they further aggregate and they further aggregate and they produce beta amyloid senile plaques. And these beta amyloid senile plaque, what they do is that they block the junction. And because they block the junction, the neurotransmitter that has been released is unable to bind to its receptors. And this prevents the neurotransmission, which results in neurodegeneration and death of the nerve cells. Apart from this, as we have already discussed, tau protein is found to be present, it is found to be associated with the microtubules. Phosphorylation of tau proteins results in its detachment, and once the tau protein detaches itself from the microtubules collapse, and there is a disruption of the intracellular transport process, and the phosphorylated tau proteins, they aggregate together to form neurofibrillary tangles. And thus in Alzheimer's disease, beta amyloid senile plaque, they block the neuronal junction. These senile plaques, they block the neuronal junctions. Neurofibrillary tangles destabilize microtubules and disable intracellular transport. So because of the phosphorylation of tau proteins, there is detachment of the tau proteins, which results in the collapse of microtubules. And because of the collapse of microtubules, the intracellular transportation within the nerve cell, it is disabled. And further, there is neuronal apoptosis, that is the death of the nerve cell. And this death of the nerve cell is caused because of the increased permeability of nerve cells to the calcium. And now we will talk about the third hallmark of Alzheimer's disease, that is a dysfunctional cholinergic neuron. Now in this diagram, I have shown two diagrams. This is the presynaptic nerve cell, this is the cholinergic nerve cell, and this is a post-synaptic nerve cell. Now this is the cholinergic nerve cell because in this acetylcholine is produced. Acetyl coenzyme A combines with choline in the presence of choline acetyl transferase to produce acetylcholine. And when the cell undergoes depolarization, there is release of acetylcholine. Acetylcholine binds to its receptors on the post-junctional membrane and produce the post-junctional activity. After that, the action of acetylcholine is terminated by an enzyme called as acetylcholine esterase. Acetylcholine esterase breaks acetylcholine into choline and acetate. Choline is not disposed of from the body, but it is taken back by the cholinergic neurons. But what happens in the Alzheimer's disease is this, that there is reduced uptake of choline by the cholinergic neurons. Uh, there is reduced availability of choline acetyltransferase, and because of which reduced synthesis of acetylcholine is there. And since acetylcholine is not synthesized in the required amount, there is reduction in the release of acetylcholine. There is a deficiency of acetylcholine, and that inhibits or that reduces the neuronal signaling. So this is the third hallmark of deficiency of acetylcholine, is the third hallmark cause of the Alzheimer's disease. Now this is a pathological cycle of Alzheimer's disease where all the three hallmarks are found to be present because of the amyloidogenic pathway. There is excessive synthesis of beta amyloid protein, and this beta amyloid protein, it forms senile beta amyloid plaques, and these plaques, they block the neuronal junctions, and which are responsible for the clinical manifestation of Alzheimer's disease, that is impairment of memory and cognition. And this beta amyloid protein, it causes neuronal degeneration. There is dysfunctional cholinergic neurons, reduced synthesis of acetylcholine, and again there is a reduction in the neuronal signaling, which is responsible for the Alzheimer's disease. And apart from this, beta amyloid proteins are also responsible for the phosphorylation of tau and formation of the neurofibrillary tangles, because of which there is collapse of microtubules and intracellular transport within the nerve cell is hampered, and thus all these are responsible for the clinical manifestation of Alzheimer's disease, that is dementia, characterized by impairment of memory and cognition. Now this slide talks about the stages of Alzheimer's disease. There are three stages of Alzheimer's disease: mild, moderate and severe. Now the disease progresses from mild to severe over a period of about 5 to 10 years. Now mild Alzheimer's disease is characterized by the short-term memory loss, forgetfulness, difficulty in recalling recent memories. Recent memories are lost first, and because of the forgetfulness, because of the short-term memory loss, there is social withdrawal, there is loss of sense of humor, and followed by this there is difficulty in learning, difficult to find words, and therefore there is language deficit. Cognitive impairment results in difficulty in thinking, impairment of planning, inability to pay attention, inability to judge, and impairment of the ability to solve problems. And short-term memory loss is followed by the long-term memory loss, and this all results in confusion. And apart from this, there are behavior and personality changes also seen in the patient. And severe Alzheimer's disease is characterized by the impairment of memory and cognition. Apart from that, there is also loss of motor skills, because apart from hippocampus and the cerebral cortex, other areas of the brain are also affected because of the progressive neurodegeneration and death of the nerve cells. There is change in the behavior of the patient. The behavior becomes very hostile. Sleeping disturbances are caused, and because of the loss of motor skills, patient is unable to talk. There is difficulty in swallowing. The patient loses the control on the process of excretion, which results in incontinence, and patient is unable to move, and the person remains bedridden, and which results in infections and bed sores, and the disease progresses from mild to severe over a period of 5 to 10 years, depending upon the rate of progression of the disease. Now we'll talk about the treatment of Alzheimer's disease. The treatment options are very limited. And one important thing over here is one important thing to note over here is this, that these options or these drugs, they provide temporary relief of symptoms, and they do not stop, or they actually do not slow down the process of neurodegeneration or the death of the nerve cells. Now, first category of drugs are the cholinergic activators, and under this category we have drugs like donepezil, galantamine, rivastigmine. All these drugs are the cerebral anticholinesterase inhibitors. Now this is the diagram that shows presynaptic neuron and postsynaptic neuron. This is a cholinergic neuron. When this nerve cell, it gets depolarized, the release of acetylcholine. Acetylcholine binds to its receptors, and after that acetylcholine is degraded by the enzyme acetylcholine esterase. Now what these drugs do over here is this, that since there is deficiency of acetylcholine, these drugs, they inhibit the enzyme acetylcholine esterase, and because of the inhibition of this enzyme, acetylcholine is not broken down and there is potentiation of the action of acetylcholine. So these drugs, they improve the cholinergic transmission, but these drugs, they provide only temporary relief of symptoms. They do not actually slow down, or they do not stop the process of neurodegeneration, progressive neurodegeneration. And the mild side effects are the nausea, vomiting and diarrhea. And these drugs, they can produce potentially serious side effects also, like slow heart rate, that is a slow heartbeat, muscle pain, and weird dreams, that is a strange and the unusual dreams. And many a times it has been seen that the drug is withdrawn because of the less efficacy and potentially serious side effects. Another category of drug is a glutamate NMDA receptor antagonist, for example memantine. Now these NMDA receptors, these are the inotropic receptors. When glutamate binds to these receptors, there is opening of calcium channels and there is influx of calcium ions in the nerve cell. Now very important thing to note over here is this, that as we have already discussed, that there is accumulation of beta amyloid proteins at the neural junctions. Now apart from blocking the neuronal junction, these beta amyloid proteins, they also causes increase in the extrasynaptic glutamate levels, and because of the increase in the glutamate level, there is increase in the calcium influx, and increase in the calcium influx within a nerve cell results in the neuronal death. Now how it all happens, let's see over here. This is a excitatory neurotransmitter. Glutamate is found to be present in the synaptic vesicle. When this excitatory nerve cell, it gets depolarized, there is a release of glutamate, and glutamate binds to its receptors, that is the NMDA receptors. There is opening of calcium channels, influx of calcium ions. Now what happens in this case is this, that beta amyloid proteins, they prevent the or they reduce the reuptake of glutamate by the glial cells, and they also cause increased release of glutamate from the glial cells. And because of the increased availability of glutamate, there is potentiation of the glutamate action, and this causes excessive influx of calcium ions. And because of the excessive influx of calcium ions, there is neuronal death. And therefore this drug, that is memantine, it is a glutamate and NMDA receptor antagonist. It blocks these receptors and it prevents the excessive influx of calcium ions. Thereby it tries to stop the neuronal death. So this drug, they reduce the glutamate transmission. However, they are also, even though they successfully reduce the glutamate transmission, they are found to only provide temporary relief of symptoms. There is no relief in the progression of disease. There is no slowing down or stopping of the process of neurodegeneration and the neuronal death. However, these drugs are better tolerated than anticholinesterases, and they show mild side effects like the tiredness, constipation, headache, dizziness and drowsiness. So there are limited options, and none of the drug is able to provide a good efficacy. All the drugs, they provide only temporary relief of symptoms, and therefore the research is going on, and the research is going on to find out the newer options for the better efficacy and reduce side effects. Currently, a number of researches are going on the experimental drugs, and these experimental drugs, they are trying to target the root cause of disease. One of the target is the beta secretase enzyme, so that there can be prevention of the formation of beta amyloid protein, followed by the formation of senile plaque. Another area of the research is the monoclonal antibodies specific for the beta amyloid proteins, which can enhance the clearance of beta amyloid protein from the body. Another area of research is the compounds that prevent the aggregation of tau. And another option is to prevent the disassembling of the microtubules. So a number of researchers are trying to develop drugs which can slow down the process of neurodegeneration, and hopefully researchers will come out with a drug with good efficacy and reduce side effects, which can actually slow down the process of neuronal degeneration and neuronal death. And thank you so much for watching this video.